So today's lecture will deal with physiologically active amines. That is the bioactive amines, what they are, and the biosynthesis of these bioactive amines. So as per the syllabus, these are the topics that are covered in Unit 4 regarding the bioactive amines. These bioactive amines, which are derived from amino acids, include neurotransmitters like epinephrine, norepinephrine, dopamine, serotonin, and GABA. As well as it also includes hormones like melatonin and histamine. And the amino acids that give rise to these bioactive amines, it can be either aromatic amino acids like tyrosine, tryptophan, giving rise to the various monoamine neurotransmitters, or it can be glutamate giving rise to GABA or gamma aminobutyric acid, then hormones like histamine derived from histidine and melatonin derived from tryptophan. Coming to the first set of bioactive amines, that is the catecholamines, which are nothing but monoamine neurotransmitters. They are also called as catecholamines because these are organic compounds that contain a catechol. Catechol is a benzene ring with two hydroxy side groups. And in catecholamines, you have a catechol plus a side chain amine group, as you can see in the structure of dopamine, norepinephrine, and epinephrine. So coming to the biosynthesis of monoamine neurotransmitters, epinephrine, norepinephrine, and dopamine. These are biosynthesized using tyrosine as the substrate. So the reactions are shown here in this box, wherein the first reaction, tyrosine to L-dopa, is catalyzed by tyrosine hydroxylase. This is followed by a decarboxylation event by aromatic acid decarboxylase or dopa decarboxylase, whereby dopamine is produced. This dopamine is converted to norepinephrine by a hydroxylation event catalyzed by dopamine beta hydroxylase. This is followed by conversion of norepinephrine to epinephrine, and the methyl group in epinephrine is obtained from S-adenosyl. So as you can see, the biosynthetic pathway of the monoamine neurotransmitters is shown here. Tyrosine, as you know, is derived from phenylalanine by a hydroxylation event catalyzed by phenylalanine hydroxylase. Now this tyrosine is the first substrate for biosynthesis of the neurotransmitters, that is the monoamine neurotransmitters. So it is hydroxylated so that you get dihydroxyphenylalanine, and this is what is DOPA. Now this DOPA is decarboxylated by a DOPA decarboxylase that produces dopamine. Dopamine is then again hydroxylated, so that yields norepinephrine. Now this norepinephrine, a methyl group is transferred from S-adenosylmethionine, which is catalyzed by phenylethanolamine N-methyl transferase, and it yields epinephrine. Now, serotonin, it is another monoamine neurotransmitter, and this is synthesized from L-tryptophan. So serotonin is nothing but it is 5-hydroxytryptamine, and this serotonin, it is also called as the happy hormone because this is a key hormone that stabilizes mood and evokes feelings of well-being and happiness, etc. So that any lack of serotonin, it is often associated with anxiety, depression disorders, etc. Like Alzheimer's, Parkinsonism, etc. So the biosynthetic pathway of serotonin from tryptophan is shown in this slide, wherein tryptophan, the first hydroxylation event of tryptophan by tryptophan hydroxylase yields 5-hydroxytryptophan, and this needs tetrahydrobiopterin as cofactor. This is followed by a decarboxylation catalyzed by 5-hydroxytryptophan decarboxylase requiring pyridoxal phosphate as cofactor, and that yields 5-hydroxytryptamine or serotonin. So this serotonin, as already discussed, it's a brain neurotransmitter causing smooth muscle contraction, and an overproduction of serotonin can cause hallucinations. So often for serotonin deficiency, vitamin B6 or pyridoxine is recommended as a supplement because of the key role in the synthetic pathway as a cofactor to 5-hydroxytryptophan decarboxylase. Serotonin can also serve as a precursor for another hormone, that is melatonin. Melatonin, as you know, it's a sleep inducing molecule which plays a key role in regulation of circadian rhythm. So if you look at the biosynthetic pathway, it is the same till serotonin biosynthesis starting from tryptophan. That is, it involves the following events. First is hydroxylation of tryptophan to produce 5-hydroxy tryptophan. This is decarboxylated to 5-hydroxytryptamine, that is serotonin. Now from here, serotonin is acetylated so that you get N-acetyl serotonin, and this is followed by, so this is catalyzed by a serotonin acyl transferase, and this is followed by methylation of N-acetyl serotonin. So this is catalyzed by the enzyme hydroxyindole O-methyl transferase, and S-adenosylmethionine serves as the methyl group donor. So this results in production of melatonin, which is N-acetyl 5-methoxytryptamine. So the next neurotransmitter generated from amino acid is GABA, or gamma aminobutyric acid. Now GABA is referred to as a non-protein amino acid, and it is an inhibitory neurotransmitter because it blocks or inhibits certain brain signals and thereby decreases the activity of the nervous system. So coming to GABA synthesis, it prominently takes place by the GABA shunt. So it is shown in this slide. That is glutamate, which can be converted to glutamine or alpha ketoglutarate, like two reversible reactions which we already discussed in earlier classes. So from this, glutamate is shunted towards GABA synthesis, and this reaction is catalyzed by glutamate decarboxylase. As you can see, it is a irreversible reaction, and it is shown in this box. So there is alpha decarboxylation of glutamate catalyzed by glutamate decarboxylase, resulting in formation of GABA or gamma aminobutyric acid. And PLP, or pyridoxal phosphate, as you can see, it functions as a cofactor for glutamate decarboxylase. So another minor GABA biosynthetic pathway is starting from putrescine, which is generated from ornithine. So in this is actually a two-step reaction, that is putrescine to GABA, and the first enzyme is diamine oxidase, converting putrescine to GABA aldehyde, followed by aldehyde dehydrogenase, converting GABA aldehyde to GABA. So as you can see, the main conversion is by synthesis is via the glutamate shunt, and this is a minor pathway for GABA synthesis starting from putrescine. So GABA is a major inhibitory neurotransmitter, and if you take glutamate, it is an excitatory neurotransmitter. So stimulation of neurons by GABA is primarily mediated by enhancing permeability to the chloride ions. So for example, drugs like benzodiazepines or Valium, it enhances the membrane permeability to chloride ions by GABA. And then there is this drug gabapentin. It actually protects against glutamate excitotoxicity, and it is an anti-convulsant medication which is used as an adjunct treatment for epileptic seizures. Now the next bioactive amine is histamine. It's a hormone, and it mediates a wide variety of physiological and pathological responses such as inflammation, gastric acid secretion, neurotransmission, immune modulations, etc. So histamine, it is synthesized in the Golgi apparatus of mast cells and basophils by enzymatic decarboxylation of histidine as shown here. So histidine decarboxylated by histidine decarboxylase, which requires PLP or pyridoxal phosphate as cofactor, generating histamine. Now histamine, as I said in the earlier slide, it mediates a range of physiological responses, and these are largely mediated through receptors, that is G protein coupled receptors. So these receptors, you have different types. You have H1 receptors, H2 receptors, etc., depending on their location. So one type of response, for example, is allergic response mediated through H1 receptors, which are found in gut, bronchi, etc. So stimulation will lead to smooth muscle contraction and vascular permeability. So antagonists have been developed to these H1 receptors. Examples include claritin, Zyrtec, etc., which are in fact second generation antihistamines. So you must be aware of the first generation antihistamines like Benadryl, which actually induced heavy drowsiness. So consequently, the second generation antihistamines have been developed, and these are the ones like Claritin, Zyrtec, etc. So besides allergic responses, histamines are also involved in other physiological responses such as control of acid secretion in stomach. So these are mediated through H2 receptors. So stimulation of through these receptors, it increases the hydrochloric acid secretion or HCl secretion. So there are H2 antagonists have been developed like cimetidine, ranitidine, etc., which are H2 antihistamines. And these H2 antihistamines are used as drugs to relieve gastrointestinal conditions such as peptic ulcers, etc. Then these H2 receptors are also found in the heart, wherein stimulation through these receptors increases the heart rate. So now we will take the gaseous signaling molecule, that is nitric oxide. Uh, so this nitric oxide, it's a key biological messenger in vertebrates, and it plays a very important role in signaling in various biological processes. So coming to its biosynthesis, nitric oxide is produced by a group of enzymes called as nitric oxide synthesis. So these enzymes, they convert arginine into citrulline, and during this process, it is shown here, that is arginine to citrulline, and during this process, as you can see, nitric oxide is produced. So the enzyme requires NADPH and oxygen serves as cofactors. Now there are different types of nitric oxide synthesis. So these have been classified according to their activity or according to the tissue type in which they were first described. So the isoforms of NOS include the neuronal NOS or the NNOS. Then there is this endothelial NOS or eNOS, and then there is this inducible NOS or iNOS. So as you know, nitric oxide, it was first identified as the endothelial derived relaxing or releasing factor, that is EDRF. And this NO is produced by many cells in the body, and particularly by vascular endothelium. Its production is very much important for regulation of blood flow. So it also acts as a potent relaxant of the peripheral vascular smooth muscle. As I said earlier, it functions as a very important biological messenger, functioning as a signaling molecule, and it has a very short half-life in the cell. Then it is lipid soluble. It is very small, so it can easily cross cell membranes. So these two features actually make it ideal for paracrine as well as autocrine signaling. So paracrine, as you know, it is like between adjacent cells, and autocrine, that is within cell signaling. So because of these features, it is ideal for paracrine and autocrine signaling. And also it does not require any receptor for its action. So it has different functions like neurotransmitter, vasodilation, bactericide, etc. So at high concentration, it has bactericidal action, and also it fights against cancer metastasis. And at low concentration, it helps in regulating the circulatory and the central nervous system. So this graphical image, it shows the biosynthesis of NO from arginine, that is conversion to citrulline. So NO is released. So the enzyme eNOS, that is in the endothelium, so it is eNOS or endothelial NOS. And then one of its function is relaxation of vascular smooth muscle. So here, as you can see, it activates guanylate cyclase, so that facilitates conversion of GTP to cGMP, and this mediates relaxation of the vascular smooth muscle cells.